Hello everyone. Today I'm going to be talking about stress and how bad it is. So, we are grateful to present to you all the hard work that our group has put into this project. We're going to delve into how chronic stress exposure can change your brain. The title of our research project is as follows: The influence of olfactory stressors on anxiety-like behavior and amygdalar brain-derived neurotropic factor, or BDNF levels, in CD1 mice. For the background information, so as you guys know, soon after birth, mice are immediately scared of cats. It's well known, a lot of people know this. So at the slightest detection of a cat, mice tend to display very stereotypical defense, freeze, or flight behaviors that indicate that exposure to cats serve as a stressful stimulus, leading to certain anxiety-like physiological and behavioral responses such as hiding, grooming, squeaking, and different things that you guys have probably witnessed before. All right. And then throughout our research and experimentation, we found that anxiety-like behaviors can be detected using tests like, as you can see, the elevated plus maze and the light dark box. So at their baseline, mice naturally exhibit curious behaviors. They like to go out and discover things, and under stressful conditions, mice exhibiting anxiety do not display curious behaviors. They instead are kind of reserved, and they don't go out and discover or explore their environment. So these tests kind of measure their anxiety and their tendency to go out and discover a novel environment. So in the light-dark box on this side, we place the mouse in the dark side and measured their behavior for 5 minutes. And the fraction of the time spent in the light versus dark side is measured at the end. And the more time spent in the light side means a much more curious, less anxious mouse. And then similarly in the elevated plus maze, we place each mouse in this middle chamber, and then measured after 5 minutes, and we measure how much time they spent in the open arm versus how much time they spent in the closed arm. And the open arm activity, or the time spent in the open arm, tells us how much anxiety they have. All right, so then this is just a picture of a mice brain, or mouse brain, and it shows the amygdala, and that's what we're going to be measuring in this experiment with the BDNF. So long-term exposure to stress causes the presence of anxiety-like behavior over time, and we see that long-term stress exposure is associated with decreased BDNF levels in the amygdala, and this part of the brain is responsible for fear and stress responses, as some people may know. So this brain-derived neurotropic factor, or BDNF, helps to increase communication within neural tissue by increasing connections between neurons. And it is involved in nerve growth and changes in long-term structure of the neurons. And it's a molecule that will largely fluctuate in levels over a long period of time in response to the chronic stressor. So BDNF is found within inhibitory neurons in the amygdala, and so less BDNF means less inhibition in the amygdala. That's why when mice are anxious and stressed out, the amygdalar tissue is overactive, and thereby correlated with a decreased level of BDNF.
Hey, Alani here. I'm going to quickly go over our methods of experimentation. Although studies have established a link between cat odor and anxiety and BDNF levels, the relationship between long-term urine exposure and the amygdala BDNF has not yet been well researched. Our research will have an intermittent cat urine exposure, and this will serve as the independent variable. And each mouse's anxiety-like response to this chronic stressor will be tested both behaviorally and physiologically. This result, both the behavioral and physiological, will serve as the dependent variable. This right here is the timeline that our group followed. Now, the first day we got the mice. We organized the 8 CD1 mice into 2 individual housing cages. This is the housing condition. The mice were kept here, which is their home, if you see right here, for a 3-day acclimation period, prior to us starting our first olfactory stimulus exposure. One cage is the control cage, and this mice were exposed to the neutral vanilla scent while the other cage is the experimental stress cage, and they were exposed to an equal amount of soiled cat litter from the same cat repeatedly for 3 weeks. Both housing cages are kept under identical conditions as you see. They both have the water, everything is identical. Alright, so after the 3-day acclimation period, the mice will be subjected to their first olfactory test for about 15 minutes, and this exposure will continue for 21 days every 3 days. Now I'm going to hand it over to Bradley to discuss our olfactory exposure procedures.
Now, in order to properly conduct the olfactory stimulus exposure, the control mice were placed into a new plexiglass cage as shown here. We placed about 2 to 3 inches of bedding in there with no food or water, and this nice little empty jar right here. Right now it's filled with cat litter, unfortunately. We then took the cage to the 4th floor computer lab in Jepson. They were allowed to get acclimated to the environment for about 15 minutes. After 15 minutes, the jar was filled with about a cup of bedding and 4 drops of vanilla extract. And each mouse's behavior was recorded. They were then placed back into their housing cages, and both the test cage and jar were cleaned for the following week. Now, for the experimental group, the same steps were taken as mentioned before, except we placed a cup of urine soiled cat litter into the jar. Before we put it back into the cage for 15 minutes, we then recorded mouse behavior after the 15 minutes. The mice are then placed back into their original cages, and the plexiglass cage and the glass jar are both cleaned. After the 21-day period of intermittent olfactory stimulation exposure and such, we then placed the mice through 2 behavioral tests, LDB and APM, light dark box and elevated plus maze, with a day of olfactory stress exposure in between them. We then killed the mice with using cervical dislocation. Control mice. Now let's talk about what we see the first time we introduce the mice to their respective experimental conditions. Now that we placed the jar with vanilla extract into the control cage, we noticed that the mice did not alter the behavior much. They were still doing the same thing. They were curious and moving around it. And after this placement of the olfactory stimulus, they continued to move around the cage as seen here. I mean, doing their own thing. Now, experimental mice. These little guys are scared. As for the experiment, we noticed that they were initially active, but as soon as the jar placed with cat urine was placed, we saw some incredible results. Just look at them go. One particular mouse immediately to the back of the jar, sat down, and he didn't move for about 3 minutes at all. He was just rigid. The remaining mice all tried to bury a hole under the jar itself, as seen here. These set of results are usually seen with every experimental procedure, and after about 3 or 4 procedures, the experimental mice would begin showing these behaviors upon being placed in the cage alone. Not even with the cat litter. Food consumption, the amount of food consumed by all the mice, had a significant increase in the food consumed each week throughout the course of this experiment. Body mass. Mice in both groups had a significant increase in their body mass, as seen here. Throughout the course of the experiment, the cumulative weight of the experimental group was significantly higher than that of the control group. Additionally, there is a significant interaction factor present between the change in body mass and the presence of olfactory stress, where experimental mice gained weight faster than that of the control mice.
Let's go ahead and go on to the behavioral results that we found. So Bradley did a wonderful job introducing us to how the mice over the course of the experiment had differences in body mass and food, but let's talk about the behaviors that they showed, specifically anxiety-like behaviors. So, as you recall, Alani and Mary had already introduced a little bit about the elevated plus maze test. So remember, in this test, a mouse that spends a lot of time on the open arm is pretty relaxed and not so much anxious, right? So in this case, we found that our mice that had been exposed to cat urine, as opposed to the vanilla extract, had spent a significantly less amount of time on the open arm of the EPM. So what does that mean? That means that these mice were not displaying normal exploratory relaxed behaviors. Instead, they were more anxious and spending more time in the closed arm. So behaviorally, that means that cat urine exposure did cause a significant decrease in their exploratory behavior and thus a significant increase in their anxiety-like behavior. Studies. So in the light dark box, we can clearly see over here again that cat urine exposure mice had spent a significantly less amount of time on the light side of our light dark box apparatus as compared to the ones that were exposed to vanilla extract. So what does that mean again? It means they were having significantly higher amounts of anxiety and less amounts of normal exploratory behavior. In case you can tell that the mice that had been exposed to cat urine and the mice that had been exposed to vanilla extract had no significant difference between their amygdala BDNF levels. Even though this is less than the vanilla extract mice, they didn't have any significant difference between the two, right? So what does that tell us? Now, amygdala BDNF levels, this is a strange finding, okay? Because when we found BDNF physically inside the amygdala, we found it from a sample of the entire amygdala as a whole. Now, we love Dr. Waters, but when he was extracting the amygdala out of our mice brains, he may have taken some residual hippocampal tissue, some other cortical tissue around it. We have no way of proving that this was just amygdala tissue on its own. However, we wanted to still kind of see, does BDNF have any correlation overall with the type of behaviors that were being displayed by our mice? And as you can see over here, we found out that mice that had spent more time in the open arm of the EPM test had overall greater amounts of BDNF inside their amygdala as a whole. Not only was this result significant in the EPM, we found the exact same results with the light dark box. Greater amounts of the amygdalar BDNF concentrations were significantly correlated with greater amounts of time spent in the light side of the light dark box. Now, the overarching message behind our research was that we found that the exposure to predator odors will exert anxiety-like behaviors in mice as evidenced by not only their eating habits, their exploratory behaviors on EPM and light dark boxes, but it has a significant correlation. This anxiety-like behavior has a significant correlation with the amygdalar BDNF levels. However, we did not find a direct connection, and we did not find that exposure altering the BDNF and the amygdalar samples of our mice. So,